Okay, good morning, everybody. So I'm back. Um, I actually was in China, so because I cannot access to the Google email, so probably I missed some of your emails, and I apologize for that. If I have not still respond, and please email me again, right? Okay, so I guess you guys had fun, right, with the amino acids metabolism session. So now we finish carbohydrate metabolism and protein metabolism, and now we start to talk about lipid metabolism. Is that okay? Everybody can hear me? Can you hear me? Okay. Oh, yes. You guys can contact ETA, to get the key and to go over your test, right? Okay, so the major lipids in the body. So a lot of lipids actually present in both plasma and tissues, and there are actually a bunch of different types of lipids. Fatty acids, free fatty acids, monoisoglycerol, diisoglycerol, triglycerol, triisoglycerol, and a cholesterol esters, phospholipids, sphingolipids, and other lipid metabolic intermediates. And all this type of lipids are present in either in both plasma and tissues. And what are the sources of the lipids in these places? Actually, two major sources, right? One is from the diet. We eat food and we absorb the lipids from the diet into the body. And the other important source is the synthesize, right? Our body can synthesize lipids. So dietary lipids and lipid synthesis are the two major sources of lipids in the body. And what are the biological roles or functions of lipids? Why we need lipids? First of all, is the energy sources, right, which is most important functions for the lipids. And it's actually a very efficient way to store triglyceride in the body. If you compare the carbohydrate proteins, you can see that 1 gram of lipids can produce 9 kcals of energy as compared to only 4 kcal of energy from 1 gram of carbohydrate or protein. And it's also very efficient to store energy in the form of fat. If you store 1 gram of fat, you use only 1 gram of water versus 4 grams of water for store 1 gram of carbohydrate or protein, right? So energy, use as energy is one of the important functions of lipids. And secondly, the structure component of membranes is also very important, right? The cell surface membrane, plasma membrane, as well as the membrane in the intracellular organelles. And this membrane structures requires the lipids, for example, the glycerolipids, phospholipids, sphingolipids, and cholesterol, all constitutes cell membranes or organelle membranes, right? And lubricants, right? This is a function way pretty easy to understand. And lastly, actually recently, I think it's very important function of lipids is signaling molecules. Some of the lipids, for example, fatty acids, can serve as the important signaling molecules and regulate the insulin signaling pathway activities, right? Regulate insulin actions. And for example, in the type 2 diabetes patients, they have higher levels of circulating fatty acids. and fatty acids can somehow stimulate or inhibits the insulin actions, so induce insulin resistance. And also lipids can be used for synthesize sex hormones, adrenocorticoid hormones, and vitamin D, right? And this type of hormones is also important signaling molecules, right? Regulates, lots of metabolic functions in the body. Now, lipid synthesis. Lipid synthesis, actually, there are two aspects, concept. One is called de novo synthesis of fatty acids. We also called lipogenesis. So lipogenesis specifically refers to synthesis of fatty acids, right? And the other type of synthesis is the triglyceride synthesis, right? So it's two different concepts, right? Try to understand, it's a different synthesis. Lipogenesis refers to synthesis of fatty acids and triglyceride synthesis. So if you still remember, this is the structure of a triglyceride, right? The glycerol backbone and 3 fatty acids attached to glycerol backbone and 3 different carbon positions, right? So triglyceride synthesis means that you need 1 molecule of glycerol and 3 molecules of fatty acids, right? And they combine, attach to the glycerol and form the triglyceride. So this process is called triglycerol synthesis. Now let's talk about lipogenesis or de novo fatty acid synthesis first. So the primary tissues for lipogenesis. So which tissues are the primary tissues for lipogenesis? Or in which tissue lipogenesis primarily occurs? Can you guys? Which tissue? Liver, yes, thank you. Liver is the most important site for lipogenesis, for fatty acid synthesis, right? So most of fatty acid synthesized in the body occurs in the liver. And there is other tissues also can synthesize fatty acids. Adipose tissue, right? So compared to liver, adipose tissue synthesize much less fatty acids, right? So liver is the primary tissue for lipogenesis, and lipogenesis also occurs in the adipose tissue, and also skeletal muscle, right? So liver is the first, the number 1, and adipose tissue is number 2, and skeletal muscle, right, number 3, and of course some other tissues like the tissues can also synthesize a little bit fatty acid, but in the normal conditions, right, these are the most important tissues that synthesize fatty acids. Okay, now the question, what do you think a high-fat diet could stimulate lipogenesis or inhibit lipogenesis? Stimulate? Are you sure? Yes? No, actually, suppresses lipogenesis, right? So keep in mind, and high-fat diet actually inhibits lipogenesis, and why, how? We will get there, right? Give explanation why. So keep in mind now this question. Okay, how about a high carbohydrate diet? Stimulates, right? We talk about a lot, right? High carbohydrate diet can cause the stimulate lipogenesis, and so carbohydrate diet is a major driver for the lipogenesis, right? So stimulates lipogenesis. So lipogenesis or de novo fatty acid synthesis may not contribute significantly to triglyceride synthesis when typical Western diets are consumed. Can you understand this? Right? Because the Western diet mostly have a higher fat, right? High fat diet. So high fat diet we know actually suppress lipogenesis. So the fatty acids that for synthesis of triglyceride mainly comes from the diet, right? If we eat a lot of, high fat diet and we actually absorb a lot of fatty acids, and this fatty acids is a major contributor to the synthesis or to the triglyceride synthesis, right? Okay, so what is the purpose of lipogenesis? Why we do the lipogenesis? What is the purpose? So it's to convert it surplus carbohydrates, for example, glucose, and protein amino acids to fat, right? And stored as triglyceride. Right? So the purpose of lipogenesis is to convert it extra or excessive carbohydrate protein to fat. And this process is occur in the liver, right? And once fatty acids synthesized in the liver, then be transported to the adipose tissue and stored in the adipose tissue in the form of triglyceride, right? So triglyceride synthesis mostly occurs in which tissue? What do you expect? Which tissue? No, triglyceride synthesis. Adipose tissue, right. Okay, so fatty acid synthesis mostly occurs in the liver, but triglyceride synthesis that primarily occurs in the adipose tissue, right? Okay, so in which tissue, in which physiological state does lipogenesis occurs? Fat condition, right? But not enough just the fat. The well-fed, high carbohydrate diet, right? When you consume a high carbohydrate diet and this lipogenesis being more active, right? Okay, so where does lipogenesis take place in the cell? Mean the cytosol, mitochondria, or other organelles? Cytosol, right? Just for the in general, most of our synthetic pathways occur in the cytosol, and oxidative pathways occur in the mitochondria, right? So synthesis or lipogenesis mostly occur in the cytosol. And what are the building block molecules of fatty acids? So what fatty acids is made of? Which building molecules? What's that? No, fatty acids. Acetyl-CoA, right? Acetyl-CoA is the building molecules for the synthesis of fatty acids. So acetyl-CoA molecule is very important, right? It's generated from oxidation of carbohydrate and amino acids, right? And when carbohydrate amino acids, too much carbohydrate amino acids being oxidized and generate a lot of acetyl-CoA, and this acetyl-CoA can be used for synthesis of fatty acids, lipogenesis. Okay. So here just sort of summarize again, for the process of lipogenesis. So the purpose of lipogenesis is to convert the surplus glucose and its metabolic intermediates and to fatty acids, and the precursors and the building block molecules acetyl-CoA, right? And acetyl-CoA can be metabolized from all these molecules, right? Glucose, pyruvate, and lactate. So the precursors are the glucose, pyruvate, lactate, right? And pyruvate can be metabolized from amino acids, right? And starting molecules is acetyl-CoA, and end product, if we're talking about the synthesis of saturated fatty acids, and end product can be palmitate. Can also be other amino fatty acids like monounsaturated fatty acids, right? And cellular location is occurred in cytosol and primary tissues, the liver, adipose tissue, and also skeletal muscle, right? And nutritional state, or in which physiologic conditions is more active, is well-fed, high carbohydrate diet, right? Okay, here I just wanted to give you sort of big picture, you know, the whole process, the lipogenesis. Again, and under the high carbohydrate diet condition, and we absorb a lot of glucose, right? So causing the high blood glucose level, which can stimulate the insulin secretion from beta cells, right? And so you have also metabolize a lot of glucose, so generate the lot of acetyl-CoA and serve as the substrate, so substrate levels of increase. And again, the liver is a primary tissue that synthesize for synthesis of fatty acids, and you know liver absorbed a lot of uptake a lot of glucose from the circulation and convert it to fatty acids, and this fatty acids process is called lipogenesis, and fatty acids need to be stored, right, in the form of triglyceride, and liver is a source of fatty acid synthesis, but not the storage of fatty acids, right? So fatty acids need to be transported to the adipose tissue for storage. And this is another process also occur in the liver, is that fatty acids first of all need to be synthesized, the triglyceride, right, and packed into the lipoproteins, right, we're going to talk about later. So lipoproteins, and the major lipoprotein synthesized in the liver is, do you guys know which lipoproteins synthesized in the liver mostly? Is that? No, the lipoproteins. VLDL, right? Very low density lipoproteins, VLDL. So VLDL, triglycerides packed into the VLDL and be transported to the adipose tissue. And this process, adipose tissue cannot directly absorb the VLDL. It's very huge, particles, right? Cannot directly absorb it. So first of all, VLDL, triglyceride in VLDL need to be break down again to the fatty acids, and then only fatty acids being able to take up by adipose cell, adipose tissue. So the enzyme called lipoprotein lipase is very important enzyme that break down triglyceride in VLDL and release free fatty acids, right, before it can be uptake into the adipose tissue. So eventually the fatty acids be taken up into adipose tissue and then re-acidify again to the triglyceride and then stored in the adipose tissue, right? So there is also processed triglyceride synthesis, in the adipose tissue. And again, the muscle, it can also take some of fatty acids and synthesize triglycerides stored in the muscle, right? So you can see the lipogenesis, the primary tissue is the liver, and the secondary tissue is the adipose tissue, and some also occur in the skeletal muscle, right? And lipogenesis is primarily, more active mostly active in the adipose tissue and also active in the liver as well as the skeletal muscle, right? Okay, now let's get to the details. Right? The synthesis of palmitate, we take the palmitate as an example, right? So see how palmitate is synthesized in the body, in the liver. So again, the acetyl-CoA is the starting molecules, right? And also need NADPH, ATP, magnesium, biotin, and bicarbonate. And the enzymes, the two key complex, enzymatic complex, are required for the synthesis of fatty acids, right? One is called acetyl-CoA carboxylase, or an abbreviation of enzyme called ACC, and the fatty acid synthesis complex, right? So for synthesis of fatty acids, a shuttle system called a citrate shuttle is very important, right? If you still remember that okay, let me just ask you a question. Where does the acetyl-CoA generated in the cells? If you guys remember mitochondria, right? Remember the one step, pyruvate being converted to acetyl-CoA, right? Catalyzed by the enzyme called pyruvate dehydrogenase, and this reactions occur in the mitochondria, right? And we also know that fatty acid synthesis takes place in the cytosol, right? So, and acetyl-CoA is the starting molecules. So how acetyl-CoA can, get into the cytosol for fatty acid synthesis? So the shuttle system, citrate shuttle system, is very important. Okay, right? So the main purpose or main function of citrate shuttle is to move acetyl-CoA cross the inner mitochondrial membrane to the cytosol for synthesis of fatty acids, right? So this is the main function of citrate shuttles. And you also still remember this TCA cycles, right? So acetyl-CoA is enter the TCA cycles and finish the first reaction in the TCA cycle and generate citrate, right? And citrate then leaves the TCA cycles, be moved out from the mitochondria to the cytosol, and this process requires the citrate shuttles. So citrate shuttles move the citrate out of mitochondria to the cytosol, and in the cytosol, citrate being then be break down to the oxaloacetate and acetyl-CoA, right? And this is the enzyme to catalyze this reaction is ATP citrulylase, right? So then you so through this system, then you move the acetyl-CoA, to the cytosol, and then be converted to marino-CoA and the fatty acid synthesis, right? And the shuttle system sort of exchange the citrate with the malate, right? So T represents the acetylate transporter, right? So they can transport the malate from cytosol into the mitochondria, which exchange with the output of the citrate, right? So the oxaloacetate being converted back to malate, right? So and for the the further exchange with the citrate, right? All right. Any question about this citrate shuttle system? Okay. Okay, so the first step also is a controlling step or rate limiting step in the lipogenesis is the marino-CoA production, right? So marino-CoA production, this is the involve the enzymes called acetyl-CoA carboxylase (ACC). So ACC is a complex. It contains the 3 different enzymes. One is the biotin carboxylase, and the second enzyme is biotin carboxylase carrier proteins, and the third enzyme is called trans carboxylase, right? So 3 enzymes constitute the ACC complex, right? And the formation of marino-CoA from acetyl-CoA also involved 2 steps, right? 2-step reactions. First reaction is called carboxylation of biotin. If you can see that the carboxyl group is actually initially from bicarbonate and being transferred to the biotin, right? So biotin actually connect to or link to the enzymes. So the first reaction is the biotin carboxylation. The biotin being the bicarbonate, the carboxyl group being attached to the biotin, and this carboxyl group initially from bicarbonate, right? And the reaction catalyzed by the 2 enzymes. One is biotin carboxylase and the biotin carboxyl carrier proteins, right? So the first reaction finished, and you see the enzyme, biotin has a carboxyl group. And the second reaction is that this carboxyl group is transferred from biotin to the acetyl-CoA molecules, right? So you add 1 more carbon to the acetyl-CoA molecules and forming the marino-CoA. So marino-CoA is a 3 carbon molecules, right? So the second step of reaction requires biotin carboxyl carrier protein and trans carboxylase, right? So again, this reaction is actually just adding the carboxyl group to the acetyl-CoA molecules, forming the 3 carbon molecules marino-CoA, and it requires 2 steps, right? Involve the 3 enzymes, and also biotin and the bicarbonate, right? And again, this is the first key reaction, right, in the lipogenesis. And this reaction occurs in the cytosol. Okay, now the next step is the fatty acid synthesis complex involved. So first I wanted to introduce this fatty acid synthesis complex. It actually the single gene that contains the 7 enzymatic activities, right? So single gene that codes the 7 different enzymes. I don't want to go each enzymes, right? And the whole complex actually is formed, is a dimers, right? Means the monomer, it has the 7 different enzymes, and the same unit, they combine together, right, and form the whole complex of a fatty acid synthesis. And they have 2 different ends, right? One is pan SH end and the cis SH end, right? And they arrange like head to tail kind of range, right? And these 2 different ends that directly involve, the synthesis of fatty acids. And before I talk about the details, I wanted to clarify the two different molecules that we talk about a lot later, right? It's called acetyl-CoA versus acyl-CoA. Acetyl-CoA is a 2 carbon molecules, right? Only have 2 carbons, and this is just a methyl group, right, attached to the CoA molecules. And acyl-CoA, you see the R group attached to the CoA molecules. The R group can be different lengths of the hydrogen carbon chain, right? So acyl-CoA has a longer CH chain, right? As compared to the acetyl-CoA, you only have one, methyl group. And malonyl-CoA is a 3 carbon molecule, right? Have a carboxyl group, the extra carboxyl group compared to acetyl-CoA molecule. So acyl-CoA molecule, sometimes we also call it fatty acyl-CoA or long chain acyl-CoA or acyl-CoA, right? So 3 different names, but just talk about the same thing, right? Okay, now it's a process of a fatty acid synthesis. It's very complicated, but you just need to know some kind of key steps, right? So first thing is first of all, this synthesis of fatty acids goes cycles. I mean repeated. Each cycles you add only 2 carbons to existing acyl molecules, right? And the first step we already talked about, right, is the formation of malonyl-CoA catalyzed by ACC, right, acetyl-CoA carboxylase. And in the first cycles, acetyl-CoA and the malonyl-CoA are required, right? So malonyl-CoA and acetyl-CoA, both molecules are attached to this fatty acid synthesis enzyme complex, right? So this represents the complex of a fatty acid synthesis, right? They have the 2 ends, right? I already told you cis SH and pan SH tail. So the acetyl-CoA molecules attached to cis ends of fatty acid synthesis complex, and malonyl-CoA is attached to pan ends of a enzymatic complex, right? This is the first step. And then the second step is the loss of a carbon, 1 carbon, right? So as the release of the CO2, so you can see the acyl molecules is transferred to the malonyl-CoA, right? So this acyl molecule initially from acetyl-CoA, right, and transferred to malonyl-CoA, and eventually form a carbonyl group. And then the next step is the reduction reaction, right? So you add the hydrogen, which initially from NADPH to the to these whole molecules, and to convert it carbonic group to the hydroxyl group, right? By adding the hydrogen. And then the next step is the dehydration. So you lose the one water molecules and generate double bonds, right? So one molecule, water molecules from this position, and you generate a double bond here, right? And then the following step is the reduction reaction again. You adding the hydrogen, which again from the NADH to the molecules and remove the double bonds, right? So you adding these two carbon hydrogen chain to the molecules, right? So the first, the cycles, you generate the one, two carbon hydrogen chain, right? Hydrogen carbon chain. So again, just this repeat, condensation, reduction, dehydration, and reduction. This reactions for the following cycles, right? So again, each cycles you adding two carbons to the existing azo molecules, so extending the carbon hydrogen chain until you reach, for example, here the palmitate has 16 carbons, right? So you go cycle to many times until you finish the adding to the 16 carbons, right? So again, the first cycles require malonyl-CoA and acetyl-CoA, and the subsequent cycles only requires malonyl-CoA, right? So the two carbon donor is malonyl-CoA, right? So each cycles you also requires two NADH molecules, right, for reduction reactions, right? Any questions about this fatty acid synthesis? Yes.
Going back to the top, there's cis and the pan.
Uh-huh.
What is pan?
Oh.
Like cis and trans?
What's that?
Is that like cis versus trans double bonds, or is that a different concept?
No, different concept. The SH, the SH, what's called a sal hydroxyl group, right? So in the different molecules, this is cysteine molecules and cysteine. Yeah, the pan. I can't remember it. Okay. I will go back to find the pan. What does the pan mean? But it's a different molecules. You have a SH group, right, attached to the different residues of the molecules. Okay. Right, is that clear for fatty acid synthesis cycles? Any question? Yeah.
You want us to know like the intermediates of each cycle, or you just want us to know that each step is like a condensation reduction and what you have written in red?
I don't think you need to remember details, each reactions, but just in general, kind of idea. I mean, what are the start molecules, substrates, and the how fatty acids is synthesized. Goes cycles. That each cycle you adding two carbons, right? And the carbon donors is malonyl-CoA, right? And just the whole process, not the details, right? Okay. So again, I just wanted to emphasize again this key reaction is still is ACC reaction that catalyzes the formation of mineral-CoA, right? And again, this process is in the cytosol. So just give you an idea, to understand better the whole process. This is the palmitate molecules, and we know it's 16 carbons, right? So fatty acids has a two ends, right? Methyl end and the carboxyl end. So the carboxyl, hydrogen carbon chain between methyl and the carboxyl end actually come from mineral-CoA, right? And the methyl and the two carbons chain actually initially comes from acetyl-CoA molecules. So in other words, when you go cycles and each two carbons, each cycle you're adding two carbons between methyl and the carboxyl end, right? Now the other thing is we know that two reduction reaction requires NADPH. Do you guys still remember one of the important pathways that can generate NADPH, and we mentioned before? Yeah, pentose phosphate pathway, right? So, so now here we'll talk about the sources of acetyl-CoA and NADPH. So acetyl-CoA basically come from the oxidation of glucose and amino acids, right? Which should be easy to remember and understand. And NADPH. So NADPH, you already told us that the pentose phosphate pathway is one of the sources of the NADPH, which is also most important sources, right? And the other two sources is from Maillard enzymes. You can see here the Maillard enzyme catalyzes the reaction, and also can generate NADPH. And then isocyanates dehydrogenase, which here is also another source of the NADH production, right? Okay, here I think is something kind of fun, okay? So calculate the numbers of, acetyl-CoA and mineral-CoA and NADPH, that how many of these molecules are required for synthesize a molecules of palmitate. Palmitate again is 16 carbons, right? So why don't you just thinking about the calculate, and we take a break, and then we come back and talk about it, right? Okay, 10 minutes break. Okay. So, counting for substrates, right? So, hopefully this will help us understand, the whole process of fatty acid synthesis. First, how many acetyl-CoA molecules are needed to synthesize palmitate, which is 16 carbons? How many acetyl-CoA is required? Remember we talked about, the those cycles, the first cycle and, and the following cycles, they actually have a different requirement, right? You said one? Oh. I don't know why. Yes, one molecule. Oh, I don't know why I put S, one molecule, right? Now, how many marino-CoA? CoA is required. 16 carbons, and each cycle we know adding 2 carbons, right? And each cycle requires 1 marino-CoA molecules. And this is 16 carbons, right? How many marino-CoAs? Yes, 7. Right? Because the first 2, methyl end carbons is from acetyl-CoA, right? And the rest, carbons is from marino-CoA. So 16 minus 2 and 14 carbons required from marino-CoAs. And how many NADPH? Each cycle requires 2, right? NADPH. And how many? How many cycles, You already told us, right? The 7, yes. So 14 molecules of NADPH. So, so just thinking about the process and how to calculate, and maybe in the test we change the carbon length and asking you a similar question, so you'll be able to calculate. Okay, so now how about the fatty acids that has longer than 16 carbons? We still talk about saturated fatty acids, right? So that means we still need the process called elongation, right? So elongation process, they occurs in the ER, and the starting molecules is the long chain acyl-CoA, right? It's different from the previous process we talked about, right? Acetyl-CoA. So long chain acyl-CoA as the substrate, and the same sequence of reactions that we talk about in the palmitate synthesis in the cytosol, right? It goes cycles, sorry, not cycle, the reactions in the whole process, like reduction, dehydration, reduction, the same process. But enzyme is different using the fatty acid elongase complex, right? And similar to the palmitate synthesis, the marino-CoA is also is a carbon donor, right? So you're also adding the two carbons at a time, right? And NADPH is also the electron donor, right? For the reduction reactions. So you're just adding more carbons depend on the length of the fatty acids, right? 18 carbons, you're just adding two more carbons, and if 20 carbons, you add like 4 more carbons or something like this. And usually, for example, the 18 carbons or 20 carbons and those, this kind of fatty acid usually are unsaturated fatty acids, right? Either monounsaturated fatty acids or polyunsaturated fatty acids. So that's why you can actually simply adding the carbon to the to the existing azo molecule because you have a double bond there. So that's why you need to go the different enzyme system, do the elongation reaction, right? So you basically elongation reaction in adding carbon to the unsaturated fatty acids, right? So for saturated fatty acids, you only able to go to 16 carbons.
So, just to clarify, because in the blue it says synthesis of saturated fatty acids greater than 16.
Okay. I sorry, maybe this is something that
Unsaturated.
yeah, usually is the unsaturated, right?
Thank you.
Yeah. Okay, so I'm not going to go to details. Okay, so the saturation, the unsaturated, the synthesis of unsaturated fatty acids, right? So it's just a simple reaction, right? Catalyzed by called a desaturase. So desaturase is an enzyme that catalyzes the formation of the monounsaturated fatty acid. Monounsaturated fatty acids only has one double bond, right? And the delta 9 desaturase is very common enzyme that present in the body, right? So the reaction is catalyzed the formation of double bond at the 9 carbon positions, right? Here. So this is the reaction. So for the palmitoyl-CoA, there's the fatty acids and delta 9 desaturase catalyzes formation of a double bond at the 9 position and forming the palmitoyl-CoA, right? So this is the unsaturated fatty acids, right? Monounsaturated fatty only have one double bond at the 9 position. And again, the delta 9 desaturase is present in the body and is very common enzyme, right? And what about the synthesis of a polyunsaturated fatty acids? Fatty acids has more than one double bonds, right? So you can see a lot of different fatty acids. It has the different lengths of carbons and also different numbers of double bonds, right? And again, it just go to elongation, desaturation, the process, right? So, and we already talked about this, right? For the synthesis of 18 monounsaturated fatty acids with oleic acid, and requires first of all desaturation, right? From the palmitate fatty acids, like we just talked about in the last slide, right? So first of all, you form the double bond at the 9 position, catalyzed by delta 9 desaturase, and forming this monounsaturated fatty acids, and then elongation, right? So you get 2 carbon more to this monounsaturated fatty acids and form the 18 carbon oleic acid, right? And similar for the other polyunsaturated fatty acids, you just go to the cycles, you're adding more double bonds and extend the carbon chain, right, to form 18 or 20 or even longer, polyunsaturated fatty acids. And you can also add the delta 4, 5, or 6, desaturates. You add a double bond at, this different positions, the carbons. Right, any questions? Okay, here just wanted to remind you that the numbering system for the fatty acids molecules, actually 3 different systems. One is numeric system, like usually use 1, 2, 3 numbers. So the first carbon, the carboxyl carbon, usually named as 1, carbon 1, and then the next carbon adjacent to the carboxyl carbon is 2 and 3, 4, right? So this is the the numeric system. It starts from carboxyl end. And also alpha, beta, and gamma system. And so the alpha carbon basically is the carbon that's adjacent to the carboxyl carbon, right? We name it alpha, beta, and gamma. And the last system is called omega system. So omega system actually started from methyl end, right? So the first carbon, the methyl end is called omega 1, right? And omega 3 and 2 and 3. So sometimes you hear about the omega 3 fatty acids or something like this, right? Depend on the position. So omega 3 fatty acid means that double bond actually at the omega 3 position, or omega 6, double bond the omega 6 carbon position. So and just try to be familiar with these different number systems. And we also talk about the beta oxidation, right, fatty acids later. So this based on the, this numbering system, alpha, beta, gamma system. So means that the break the carbon chain between alpha and beta carbon, right, when fatty acid is being oxidized. So that's why we call it beta oxidation. Okay, now regulation of lipogenesis. We already talked about it, the high carbohydrate or high sucrose diet are the most important drivers for the lipogenesis, right? And insulin is also important regulators because high carbohydrate diet can stimulate a lot of insulin release, right? So insulin is an important hormone that mediates the effect of a high carbohydrate diet on the lipogenesis, right? So insulin directly regulates the lipogenesis. And on the other side, that glucagon, which is increased in the fasting conditions, right, so inhibits lipogenesis, which makes sense. And calorie restriction and high fat diet and are the negative regulators of lipogenesis. They are all inhibit lipogenesis, right? This is just a general picture, overall regulation of the lipogenesis. And if we wanted to go to the details, the regulation, again, we always interested in the key enzymes in each pathways, right? Because the key enzyme is the place that hormones or other substrates regulates it and then control the pathway processes. And for the lipogenesis, the key enzyme is the acetyl-CoA carboxylase, ACC. If you still remember, ACC is catalyzed, the function is to catalyze carboxylation of acetyl-CoA to form malonyl-CoA, right? So this is the key reactions. And how ACC is regulated is very important that control the lipogenesis, right? At 3 different levels of regulation. First is the allosteric regulation. For example, citrate is the strong stimuli of lipogenesis or ACC activities, right? So if citrate levels increase, which occurs in the high carbohydrate diet condition, right? You know, we have a lot of acetyl-CoA being generated from oxidation of glucose and then enter the TC cycle and produce a lot of citrate. So citrate increase then can stimulate lipogenesis, right? Citrate be moved out to the cytosol by which system, shuttle system, remember? Citrate shuttle, right? To move, citrate out of mitochondria to the cytosol and for lipogenesis. So citrate is a strong stimuli of the lipogenesis. And long-chain acyl-CoA is actually inhibitors of lipogenesis. So try to connect the high-fat diet, why high-fat diet inhibit the lipogenesis to long-chain acyl-CoA, right? So in which conditions the long-chain acyl-CoA levels increase? You know, in the high-fat diet, right? When we, in high-fat diet, we absorb a lot of fatty acids into the body. So long-chain fatty acyl-CoA is increased, right? So this is the reason why high-fat diet inhibits lipogenesis, because when we consume high-fat diet and we increase long-chain acyl-CoA, which is the inhibitor of the ACC, right? Then inhibits lipogenesis. So this is the first levels of regulation. And the second levels regulation is called covalent regulation, basically phosphorylation, dephosphorylation, right? So the two hormones, that insulin and glucagon, regulates ACC enzyme activity by controlling phosphorylation or dephosphorylation of ACC, right? And last levels of regulation, the gene transcription. So insulin and glucose both can increase ACC gene expression, increase the levels of ACC, and glucagon cycle AMP decreases ACC gene expression, right? So three different levels of regulation and involve the substrate, citrate, long-chain acyl-CoA, and the two important hormones, insulin and glucagon, right? So try to remember, how ACC enzyme activity is regulated and connect this to the regulation of the lipogenesis, right? And another important thing is I wanted to introduce the enzyme called AMPK, right? The AMP activate protein kinase, and this kinase is very important and mediates the hormone regulations, right? Insulin and glucagon epinephrine. So what AMPK does is to phosphorylate the ACC, right? So once ACC is phosphorylated and the enzyme activity is actually decreased, right? So AMPK is catalyzed phosphorylation of ACC and inactivate ACC activities. And how AMPK is regulated? So AMPK is activity increased by glucagon or epinephrine, right? So during the starvation, when cellular level energy is decreased, and when glucagon and epinephrine levels increase, and AMPK is activated, right? So AMPK and phosphorylate ACC and inactive ACC. So, and decrease lipogenesis. Right? Is that clear about this process? No? So just adding one more component in this regulate process, right? You know, insulin, we know insulin regulates ACC, right? And phosphorylate, dephosphorylate ACC. So that mediated by AMPK. Sorry, I messed up. I mean the glucagon and epinephrine levels, right? So AMPK directly regulates ACC, right, by phosphorylate ACC and inactivate ACC. And AMPK is increased during the fasting, right? You know, the glucagon and epinephrine level can increase ACC and AMPK. Right? Any questions? Okay. And here just wanted to talk about a little bit more about the how long-chain acyl-CoA inhibits lipogenesis. So sort of more detailed mechanism, how, right? So first of all, long-chain acyl-CoA can inhibit acetyl-CoA carboxylate activities, right? We just talked about, So actually, long-chain acyl-CoA has multiple actions, and then they work together to inhibit lipogenesis. Right? First is inhibits ACC activities, and second inhibits mitochondrial tricarboxylate transporters. Right? So this transporter is important, right? Work together with the citrate shuttles to exchange the citrate with malate. Remember, so. So long-chain acyl-CoA can inhibit this transporter. And what happens if the tricarboxylate transporter is inhibited? If this transporter is not functioning, what happens to citrate movement? You have a less citrate movement from mitochondria to cytosol, right? Because citrate movement requires this, this transporter exchange, work together with the citrate shuttles. So if this transporter is inhibited by long-chain acyl-CoA, and you have decreased transporter citrate into the cytosol, and then preventing the process of the ACC. Remember, the citrate is the allosteric regulator of ACC, right? So if you have decreased levels of citrate in the cytosol, then you have less activity of ACC. Right? Does that make sense? Okay. And lastly, long-chain acyl-CoA can also inhibit pyruvate dehydrogenase. Pyruvate dehydrogenase is the important enzyme that catalyzes the oxidation pyruvate to acetyl-CoA, right? So if this enzyme is decreased, what happens to the substrate of fatty acid synthesis? Happen to acetyl-CoA levels will be decreased, right? So overall, that the long-chain acyl-CoA inhibits lipogenesis through the following mechanisms, right? First, they inhibit ACC activities. And second, inhibits the tricarboxylate transporters, so decrease citrate levels, right? And lastly, inhibits pyruvate dehydrogenase and decrease the acetyl-CoA production. You decrease the substrate, right, for the synthesis of fatty acids. Right. So again, for the whole thing, like how high fat diet suppresses lipogenesis, the reason is because high fat diet can causing increased long chain acyl-CoA, right? And long chain acyl-CoA inhibits lipogenesis, and the mechanisms involve this three process, right? Inhibits long chain fatty acyl-CoA, inhibits ACC activities, inhibits tricarboxylate transporters, reduce citrate levels, and inhibits pyruvate dehydrogenase and decrease the acetyl-CoA levels, right? Any questions about this process? Okay. Yeah, that's just the diagram. Just show the maybe more clear pictures of just I've already talked about it, right? You know how long chain acyl-CoA inhibits lipogenesis, right? Okay, so for the regulation of lipogenesis, and here's the summary, the insulin regulation of lipogenesis. And increase the ACC activity, increase glucose uptake into the adipose tissue. So, and insulin we know it stimulates lipogenesis, right? So, and this is just to explain more details how insulin induces lipogenesis. And first, increase the ACC activity. And insulin we know increase glucose uptake, right, in the adipose tissue. So you have increased pyruvate and increased glycerol 3 phosphate, and activates the pyruvate dehydrogenase. So you have increased acetyl-CoA molecule production, and suppresses the cyclic AMP levels. So inhibit lipolysis and decrease fatty acids release. So you have a decreased long chain acyl-CoA levels. So you have a less inhibition of lipogenesis by long chain acyl-CoA, right? Is that clear? Maybe for insulin, how insulin regulates lipogenesis. First of all, directly stimulate ACC, and indirectly increase glucose uptake, increase pyruvate levels, and which is the precursors, right? And glycerol 3 phosphate, and increase pyruvate dehydrogenase. And here may be a little bit tricky, but you can see that because insulin suppress the lipolysis, right? Lipolysis means that you have a less fatty acids released, so you have a lower long chain acyl-CoA when insulin level is high. So long chain acyl-CoA we know is the inhibitors of lipogenesis, right? So if you have a lower long chain acyl-CoA, then you have a less inhibition of lipogenesis. So the net result for insulin regulation of lipogenesis is to stimulate, right? It stimulates lipogenesis. So in general, insulin is a hormone that stimulates lipogenesis. Okay, any questions about the the concept that we talked about so far? Okay. Okay, so so this is just a summary for fatty acid synthesis or lipogenesis. Maybe we wanted to go over again, right? So hopefully we'll still. So in which physiological conditions that lipogenesis occurs? High fat diet, right? High carbohydrate diet, right? And what is the purpose of lipogenesis? Why we need to do lipogenesis? Right? Because we have too much sugar and too much protein being consumed, and then we wanted to convert this to energy, right? So fat and store in the body. So and in the cells, the fatty acid synthesis is occur in the cytosol, and which is the building blockers of fatty acid synthesis, lipogenesis. Say again. Acetyl-CoA, yes. And the two enzyme systems that involve in the lipogenesis, which one is a key enzyme for the lipogenesis? ACC, right? Acyl-CoA carboxylase complex. And what does ACC do? Yeah, to convert it as acetyl-CoA to mineral-CoA, right? To catalyze the formation of mineral-CoA. And for the whole fatty acid synthesis process, it goes cycles, right? So the first cycles, what molecules are required for the first reactions, for first cycles of fatty acid synthesis? Acetyl-CoA and monomer-CoA, right? And which molecules is carbon donors? Mineral-CoA, right? So each cycle you're adding 2 carbons to the existing acyl molecules, and the mineral-CoA is a 2 carbon donors, and also requires the electron, right? Electron donors is NADPH, right? We have 3 sources of NADPH. Can you guys tell me 3 sources? First, for NADPH, pentose phosphate pathway, right? And second, malate enzymes, and third, isocytrate dehydrogenase, right? Okay. Okay, about the regulations. So I think the important part is the 3 levels of regulation of ACC, because ACC is a key enzyme, right? So we need to understand how ACC being regulated at the 3 different levels. First level is the allosteric regulators. What are the 2 important allosteric regulators of ACC? Citrate and long-chain acyl-CoA, right? And at second levels of regulation, hormones, right? Which are 2 important hormones. Glucagon and insulin. How insulin regulates ACC lipogenesis? Increases, right? Stimulate lipogenesis. And glucagon reduces, right, the lipogenesis. And the third level is the gene expression, right? And insulin, glucagon does the opposite. Insulin stimulates ACC, transcription, and glucagon inhibits ACC transcription. And another thing is important, I wanted to connect or explain the reason why high-fat diet inhibits lipogenesis. So you connect long-chain fatty acyl-CoA, right, and to understand the, how the mechanisms, how long-chain acyl-CoA inhibits the lipogenesis, right? Involve the three mechanisms, right? Remember? So first is it directly inhibits ACC. Secondly, inhibits the tricarboxylate transporters, right? Reduce citrate levels. And third mechanism is to inhibit pyruvate dehydrogenase and reduce acetyl-CoA concentration. Okay. All right. So now let's talk about the triglycerol synthesis. So triglycerol synthesis means that the free fatty acid molecules attached to glycerol backbone and forming triglyceride, right? So this is the whole process of triglyceride synthesis. So again, it's a different concept from the lipogenesis, right? And glycerol backbone, the substrate for triglyceride synthesis is actually glycerol 3-phosphate. So at the carbon 3 position, you have a phospho group, attached to this glycerol. This is the substrate, right? So why triglyceride need to be synthesized and stored? And I think we already answered already here, right? Because we mentioned earlier the lipids like free fatty acids sometimes is important. The signaling molecules regulate the metabolic processes, right? So we don't want to have too much free fatty acids circulating around, right? And that will, and also long-chain fatty acyl-CoA is also the regulators of lipogenesis, right? So, so we wanted to the free fatty acids being attached to the glycerol backbone and form triglyceride and store in the adipose tissue. That way that we can control the levels of free fatty acids in the circulation, right? So this is an important, one of the important reasons why we wanted to synthesize triglyceride and store triglyceride in the body. And of course, the other reason is for the energy, right? In the fat condition, we have extra energy, so we store energy in the form of a triglyceride, and during the fasting, we can use the, break down the triglyceride as an energy source. So where are triglycerides synthesized? So which tissue are the main tissue for the triglyceride synthesis? Adipose tissue, right? And the liver, and also many other tissues, including muscle, other tissues. It can also synthesize triglyceride. In the cells, facing the cytosol, this place is the main site where triglycerides synthesized. And where are the triglycerides stored? A major site is adipose tissue, right? Okay, so substrates of triglyceride synthesis, fatty acids, right? Of course, 3 fatty acids and 1 glycerol backbone, glycerol 3-phosphate are the substrate of triglyceride synthesis. And before fatty acids be able to attach to the glycerol backbone, fatty acids need to be activated first, right? So this is the process, the reaction for the activation of fatty acids. So fatty acids, the enzyme is called fatty acyl-CoA synthetase, right? So fatty acids being activated to fatty acyl-CoA. So CoA molecules being attached to the fatty acids and forming fatty acyl-CoA, right? And enzyme again, the fatty acyl-CoA synthetase, and requires 1 ATP. And the other substrate is the glycerol 3-phosphate. Actually, there are two sources of glycerol 3-phosphate. One source is of glycerol 3-phosphate we talked about earlier, if you still remember, from which nutrients metabolism and which pathways, where glycerol 3-phosphate being generated. Glycolysis, yes. So glycolysis is the important pathways for generate of a glycerol 3-phosphate, right? And this is the enzyme called glycerol 3-phosphate dehydrogenase, right? Catalyze formation or generation of glycerol 3-phosphate. And another source of the glycerol 3-phosphate is the glycerol can be directly phosphorylated to glycerol 3-phosphate, right? And this reaction is catalyzed by glycerol kinase. So remember, glycerol kinase is only present in the liver, right, and not in the adipose tissue or skeletal muscle, right? So again, in the liver, glycerol 3-phosphate can be generated from two sources. One is glycolysis, and the other is directly phosphorylation of the glycerol by glycerol kinase, right? But in the adipose tissue and muscle, the glycolysis is the only sources, right? It's only sources of the glycerol 3-phosphate. So here's the question. So where do fatty acids come from for triglyceride synthesis? So first of all, in which physiological condition triglyceride synthesis occurs? Which physiological condition? Fat or fasting? Fat, right? In fat condition. So the sources of a triglyceride, the fatty acids for synthesis of triglycerides, they come from the three pathways, right? First is the diet, fatty acids from diet, and second is the body stored, and third is de novo lipogenesis, right? Because triglyceride synthesis occurs mostly in fat condition, right? So we eat the diet, and we absorb the fatty acids into the body, and these fatty acids need to be stored in the adipose tissue, right? Synthesized triglycerides. So dietary sources is one of the important sources. And then the second, the body stores. We know, we stored adipose tissue we stored fatty acids or triglyceride in adipose tissue, right? So sometimes, or in some pathophysiological conditions, for example, in the type 2 diabetic conditions, we for some not very characterized mechanisms that the lipolysis actually increase in the diabetic patients. So you have they have more fatty acids being released from adipose tissue. So you have high levels of fatty acids. So those fatty acids also need to be synthesized as the triglyceride in the liver. So under these disease conditions, the fatty acids from adipose tissue being transported to the liver and synthesize triglyceride, and triglyceride mostly being stored in the liver, so causing the fatty liver disease, right? Okay, so and the third condition is the de novo lipogenesis, right? So, for example, if we consume high carbohydrate diet, or we have increased de novo lipogenesis, right? So we have a lot of fatty acids synthesized. So here I wanted to clarify, under the normal physiological conditions, the triglycerides synthesized in the adipose tissue, the fatty acids mainly from two sources, right? Dietary lipids and the de novo lipogenesis, right? And under the normal conditions, and triglyceride synthesis is mostly occur in the adipose tissue, and fatty acids coming from diet and the de novo lipogenesis in the liver, right? And only under the pathological condition, like I described in the type 2 diabetes, because of lipolysis is increased. So you release a lot of fatty acids from the adipose tissue and then pick up by the liver and synthesize triglyceride in the liver and store triglyceride in the liver. So it's different, the process, right? And happening in the different organs, places, tissues. Okay, any questions about the triglyceride synthesis? Okay, so let's see if I have how many slides. What do you guys think? Should we finish the synthesis, or we just leave it for next time? For next time, okay. Too much information, right? Okay.